Dr. Adam Brufsky, Temple board member, and I'd read your whole bio, but I'm not sure we have enough time in this interview. You're a professor, director of the McGee Women's Cancer Program, co-director of the Comprehensive Breast Cancer Center here in Pittsburgh. Dr. Brufsky, Adam, how are you?
I'm fine, Aaron. Thank you very much, Rabbi. Thanks for having me.
I'm excited to get to have this conversation. You've been thinking a lot and writing a lot about COVID-19. So would you just take us back? What do we know so far about this virus?
Well, let me start by going back a little further. Why would a medical oncologist who does breast cancer be interested in an infectious disease? And, it goes back quite a bit. And I think it's important to talk a little bit about the philosophy, of how kind of people do stuff. Right now in medicine and everything, and we're having arguments like this right now that people really just want to do clinical trials. I mean, not only clinical trials, but we usually use a lot of science and clinical trials to help us figure out what works, what doesn't, what theories are right and what isn't. But I think what people don't understand is that clinical trials like this, happened, really only started probably in the 1950s, after the discovery of penicillin. And, we tried to test whether things are right, or there's streptomycin, tuberculosis. And what people tend to forget, in the olden days, before that, maybe 100 years ago, the last time we had a pandemic like this, the way we used to do things is that we would see things in the clinic. We make a clinical observation, and then we'd appeal to the literature. What we'd say is, okay, we saw this. Now, what does the literature say? Let's try to match it up and kind of come up with theories about what's happening with the world around us. And you come up with a theory, and it would be, oh, it may work, it may not, and then you'd adjust it. And so what happened with this is that all the stuff with hydroxychloroquine started to make the news about 6 weeks ago or something. And I realized that we work with hydroxychloroquine in breast cancer. So the whole idea is that colleagues of mine at Penn have been working with it to try to clear breast cancer cells out of the bone marrow. So one of the things we think happens in breast cancer is that, you have a breast cancer and then it comes back like 3 or 4 or 10 years later, and we wonder why. And one of the hypotheses is that little cancer cells break off from the breast and sit in your bone marrow and live there for many years and then grow back and cause you problems. And we call that as tumor dormancy. And so I was interested in hydroxychloroquine for that reason. I've been kind of not participating in that particular trial that's at the University of Pennsylvania, but we've all been interested in this problem for many, many years. And it got me thinking about hydroxychloroquine. And it turns out one of the things hydroxychloroquine does is that it's a hypoglycemic agent. It's like metformin. In fact, in India, when metformin was still on patent, they used to use it all the time. You can't use it for a long time because, as people have found out, you get retinal problems, you have occasional problems with your heart. So you can't really use it that often in this syndrome. But anyway, so there was that. And then I was talking to some guys and, who were seeing COVID patients. And we just happened to be discussing it. And they go, Adam, these patients not only have diabetes, because diabetes is a risk factor for it, but they also have really high blood sugar. They have unexplained diabetes. We don't know why. They never had diabetes before. And so, again, as the docs of old did, I went back to the literature and I found that they had this model of mice. And in the mice, these are mice that are diabetic. They're called NOD mice. And when they're diabetic, they have really high levels of ACE2 in their lung. ACE2 is the receptor for the virus. And when basically they gave them insulin, the levels went down. But it turns out it wasn't the actual levels of ACE2. It was a level of ACE2 with sugar added to it, what we call glycosylated ACE2. And so it was really interesting. And I said, this kind of made sense. And I started looking into some other stuff and experiments that they've been doing in monkeys who had SARS. And, we looked at SARS because SARS is really what is most similar to COVID-19. They're very similar diseases and they're pretty much very similar viruses. And so I started to look into it and I came up with this idea that basically the disease could really be explained by the amount of virus with sugar on it, the amount of receptor, the ACE2 receptor with sugar on it, and then the amount of immune response you had about a week to 10 days into it, that would determine your symptoms in your clinical course. Because what would happen if you had a lot of virus still sitting around there with a lot of sugar on it, you had a lot of sugar kind of on the proteins of your immune system and a lot of sugar on the receptor, the response would be huge. You get the cytokine, what they call the cytokine storm, and you get ARDS. You'd have to get a tube down your throat, need a ventilator. And it really seemed to be all related to that. And so I wrote a paper on that was published. I guess now this field is going so fast, around the 12th or 13th of April. And then I looked around and I said, okay, the next thing you got to say is, okay, we have this going on, this issue. What would, maybe we'll look at the virus to see if there are mutations in the virus itself, because this virus being an RNA virus, it mutates a lot through time. And these guys in Europe, this German group actually has a way of tracking all the DNA sequences of the virus. So we always do the sequence of it through
So
time.
let me jump in real fast, just to ask a point of
Ask whatever you want.
Right, so there's a lot of the understanding when this was first coming out, or thought we understood, was that some of the comorbidities were around smoking or other lifestyle choices. And you're suggesting
We'll get to that. Yeah.
Blood sugar is really, really prevalent in why this is such a health risk for people.
Yeah, well, it turns out 3.8% of the US population has undiagnosed diabetes. They don't know it. You know, we check it through something called hemoglobin A1C, and everybody probably knows what this is because you go to your doctor and they do it all the time. And every time you come in for your annual physical, you get a hemoglobin A1C, and that says whether you have diabetes or not. And so you want that number to be below 5.4%. And it turns out they did a study and they found like 3.8% of the US population without a history of diabetes randomly had it. And, you know, so this is something, it's easy lifestyle choice, exercise, diet, make sure your hemoglobin A1C is low, and it'll protect you from COVID probably, but other stuff too. You know, even if it doesn't protect you from COVID, it's a good lifestyle choice regardless. And I think that's the point of the paper. So anyway, so then you want to go on? You want to have more questions?
Please, yeah. Tell me about this mutation and some of the things.
So what happened is, what happened is that I went around looking, you know, saying, okay, if this is really true, then maybe there are mutations in the virus itself because it mutates all the time. You know, maybe there are mutations in the virus that would determine whether it's more aggressive or less aggressive, depending on the amount of sugar on its coat. And so these guys from Germany, they put this called GISAID. You can sequence the virus, like they stick this thing up your nose and they do the viral sample, but what they can do with it is they can actually do DNA sequencing on it really fast. And what we couldn't do back 15 or 17 years ago in SARS is we didn't have this technology. In 2020, we have this technology and we literally can do it that day. It takes a couple hours and put the sequence right up on the internet for people to analyze. And so there are these guys from another group called NextStrain that you can do it. Anybody's open source. You go to nextstrain.com. You can play with it yourself tomorrow. If people want to do it, it's really kind of interesting. And you can see where the virus has gone and what mutations it has and where they go. And what happened, it was a really simple thing. I'm not the only one who did this. There's lots of groups around that did it. But one thing I noticed is that there was a particular mutation that seemed to arise in China where the virus started, and it changed one amino acid to another, right in the sugar in the coat of the virus, what we call the spike protein. And it turns out that this mutation, at least in theory, should increase the amount of sugar molecules around where that virus is, or where around the viral protein where it is. It should increase what we call glycosylation of the virus. And so it was really interesting because what happened when you look really carefully, and again, there's another paper that we published, gosh, I'm trying to think of when that was. Things have gone so fast you forget. So it probably was around the 17th or 18th, maybe the 20th. It was about 3 weeks ago. And we wrote this paper where basically, you know, we found this, but the interesting thing about it was that the normal version, the less virulent version, was on the West Coast. And the predicted more aggressive version was on the East Coast. And when you look, you saw, wow, the virus seemed to be less aggressive on the West Coast and more aggressive on the East Coast. I think everybody knows that, right? They saw New York City, all the people dying, you know, where you go to California and it's, yeah, people are dying, God forbid, no one wants to die. But in Washington and Oregon, they really weren't dying in as great a number from the virus. And it's kind of weird in a state of 40 million people that there really are only probably maybe 1,000, 2,000 deaths probably predicted. Where in New York City, you know, the center of it all, it looks like as of today, there are about 13 or 14,000. So, you know, the thing is, why is that? And that was the second piece. And we published that a little bit before. And so then the next question, okay, was why? Why does adding sugar to this virus make a big difference? You know, that's really a question that we're always asking. And, you know, so, you know, when I started thinking about this, I got someone who's a very smart guy. He's one of the brightest immunologists around. He's a guy named Mike Lotz. He's at the University of Pittsburgh. And his expertise is in a cell called the dendritic cell. And he likes to call the dendritic cell the convenient sort of immunology. Okay, and the reason he says that is that what the dendritic cell does, it's responsible for finding all these foreign proteins in your body and sucking them up. Okay, it's called endocytosis. And it sucks them up and it processes them and then presents them to the immune system. It dissolves them up. So all these proteins, suddenly your immune system, your T cells and your B cells, they all see it and they go, oh, okay, this is what we need to react to. So the dendritic cell is really central to the entire immune response. And it's central to a lot of different things, these dendritic cells are in your skin, they're in your gut, they're in your everywhere. They're all over your body, but they grow in your bone marrow and they migrate all over your body. Anyway, it turns out when we looked at SARS, again, which is the most similar thing to SARS-CoV-2, right? It's like COVID-19. It turns out that the virus needs to, how can I say this? It gets endocytosed, it gets sucked into the dendritic cell and it lives there. Because there's another, dendritic cells have the ACE2 receptor on them. You'd expect them to have that. But it turns out they have another receptor on them called DC-SIGN. And all the cancer cells all over your body, the lung cells where you'd expect to have COVID-19, your brain, your nose, your heart, all these places where you see COVID-19 infection, pancreas, they have this, it's called L-SIGN. It's the same kind of receptor, but it's called DC-L-SIGN. And it turns out that this receptor is responsible for attaching to bacteria and viruses that have sugar coats, and with the ACE2 receptor, sucking them into the cell. And so it turns out that if you have more sugar, this receptor will suck you into the cell better. That's the whole idea behind it. Now, the virus lives in the cell. It doesn't replicate. It kind of affects it a little bit, so it doesn't really stimulate the immune system. It kind of paralyzes it for a few days while your immune system doesn't know what to do. It kind of dissolves. Your immune system is looking around going, what am I supposed to do? The dendritic cell's not doing anything. So it's just sitting in the cell in what we call the endosome, which is kind of, you remember from high school biology, it's the Golgi apparatus, the lysosome. That's where the virus lives. And it just sits there and then it waits for the immune system to get really kind of messed up. And then it spits it right back out. It's called the viral synapse. It's kind of evil in a way. And, and then you get a really big response. If suddenly this virus is getting spit out, day 5, 6, 7, 8, suddenly, boom, your immune system tries to attack it. And so that's what causes all this ARDS and everything else that you see, the cytokine storm, et cetera. At least we think. As my wife Jill has told me, it's only a theory. Okay, and so, you got to prove all this. And again, you can't, because we're in a pandemic, and this is what you have to do. You have to go to the literature, see if what you're seeing makes sense. Like, we know all the T cells go away initially in this disease, which is consistent with the dendritic cell being paralyzed. We know at around day 8, you get the cytokine storm. It all makes sense. It's fitting with the observations. That's the key. If it didn't, we wouldn't have promoted this theory. And so that paper actually is being published actually was published on Monday. And because these things are embargoed, it's probably going to come out tomorrow morning. So, it's okay for me to talk about it now. They've had it already for 2 days. And what are we going to do? I got to talk about it now. I have the preprint if people want to see it. I've circulated it to probably 50 or 60 people already to get their opinions. And most people are pretty impressed by it. They like it. Not impressed with me. They're impressed with the idea. They think it's a kind of a cool thing. And, so what happened is that the interesting thing about this paper is that toward the end of me writing it, toward the end of us writing, because me and Mike, toward the end of us writing it, we found these mutations that are suddenly people are finding these other mutations around the world, not just this D17, D16G, but other ones. And when you look at these mutations, a lot of them predict not for becoming more aggressive. And if everybody read the paper this morning, there's a group at Los Alamos that kind of was looking at the same mutation. I've been talking to them actually for the last couple of days. They found this mutation. They go, oh no, it's aggressive, it's terrible. But what they don't say, okay, and what's going to come out in this paper is that there are mutations that make it less virulent, okay? And that's the point. Viruses get less virulent over time, and you expect them to accumulate mutations over time that make the disease less virulent. And so that's kind of what's going on. We found these mutations that predict for less, what I call glycosylation, adding sugar molecules to the virus. There are less of those mutations over time, and that's kind of what's happening. Again, only a theory, you can't use it. So, what do you do? With any theory like this, you use it for prediction. What are you going to do now? We're trying to relax social restrictions and, see how things go. If this theory is even partially correct, there's a few things that we'll find. We should start to find viruses late in the course of COVID-19, a little bit later, not like earlier when it started in China, but now, that predict for less virulent viruses. Well, it turns out this morning in Arizona, again, some guys just put this up. What we do now is we don't do peer-reviewed science anymore. I'm trying to, but they just throw these preprints up on the internet, let people look at it before they get peer review. which in some ways is good because you want to give people information. In some ways it's bad because we don't get to discuss it internally and privately before we put it out to the general public. And there's often a lot of questions that we have with the data that we prefer we discuss amongst ourselves, and get those questions answered before we throw it out there. But unfortunately, everyone wants to know. Everybody's frightened, they're scared, they want info of the unknown, and everybody's all out of the best of intentions. But often times when you don't do peer review, you can kind of get yourself into a little bit of a pickle. So the bottom line is that in Arizona this morning, these guys presented this paper on the internet where they found a mutation very similar to one was found in SARS toward the end of the SARS epidemic that predicts for attenuation of the virus. So that means that the virus could go away like SARS. There's a possibility that that could happen. Doesn't mean you should run outside tomorrow. I think what we need to do is be very cautious, wait a few weeks, see what happens when all the social restrictions are relaxed. And what we hopefully will find is that people won't get as sick. It doesn't mean the death rate. Okay, you can't look at death rates on TV or anywhere. You can't read those because those are confusing. The reason they're confusing is that people mix everything into it. If someone dies of pneumonia, I wrote pneumonia on the death certificate. Sometimes I don't know whether they're positive for COVID-19 or not. That's only sorted out later. And it turns out there's a lot of people dying of regular pneumonia, like a bacterial pneumonia. People still die of bacterial pneumonia. People still die of fungal pneumonia. They die of other pneumonias we don't even know, yet they sometimes get coded as COVID-19 without having a positive test. And so that's number one. So you can't really look at the deaths. You also can't look at the number of infections. They go, oh no, the infection rate went up. You can't look at that because you could be infected with a milder form of the virus. And the assays we have right now only tell you whether you have a virus. They don't tell you if it's mild or not. That's the second thing. So the only thing you really need to look at when you look at all these statistics, you need to look at the number of patients who are in the ICU. Is that going up or not? If it's going up, it means a severe form of the virus is going around. If it's going down, it means it's not. That's really the only statistic you can use. And to be honest with you, the only way you can get that is off the state website. And it depends on the state. I don't know if Pennsylvania has it. I know Georgia has it. I think New York has it. I think, California may have it. It really depends. You've got to go state to state, and all the state websites are available. They're just search coronavirus in the state you're interested in, and it should show up. I don't know Pennsylvania. I haven't looked at that recently, where they can tell you, they tell you the number of people in the hospital, they tell you the number of the deaths, number of infections, but they don't tell you the number of people in the ICU. So it's hard to do that. So really, that's kind of where we're going. And I think that, fingers crossed, keep our fingers crossed here. I wouldn't run out of, if I'm staying in an elder care, nursing home, or assisted living, I wouldn't get outside yet, and I wouldn't go see my kids yet. But I think that, we have something now that's testable.
Allegheny County has it.
Oh, they do? My wife just told me that Allegheny County has a number of ICU beds. Good, thank you. The peanut gallery has spoken. So the bottom line is that, so that's great. So we actually have that now, and we can look at it and we can say, okay, let's see what's happening as we all go out and do stuff. And gradually, as we kind of get more and more together, and maybe some of this distancing kind of goes away, and slowly but surely, we watch the ICU beds with COVID-19. It could be, remember, people go to the ICU, they get sick. That's not going to stop, but what we look for are people in the ICU with COVID-19. And if that doesn't seem to rise as we go and become more and more socially active again and become more human, to be perfectly frank, I think that is a good thing. And I think that's what we're looking for now. At least I am. Again, the issue with this whole thing, I'm just one person. There's a lot of docs who think like me, by the way. I'm not the only one, but this is a discussion that we all have to have amongst ourselves, amongst the physicians. It's hard to have those discussions in private before we talk to people because everybody wants to know. Under normal circumstances, we take a couple months or a couple years. We'd have meetings, we argue, we say this, you're wrong, no, you're wrong, no, this is right, no, that is right. That's how science and medicine gets done. That's how it normally happens. But we're in the middle of a pandemic, a 100-year flood in a way. And it's really hard for us, when the people want to know, to not kind of just come out there and not do it. And I think that's where we are.
Let me ask you a couple of questions on that sharing of information and on the way these papers are coming out. And then I want to return to just making sure that I understand on behalf of everybody who is joining.
Please, I know it's a huge mouthful. I've tried to make it as simple as possible. The concepts in this are kind of crazy. And in fact, the weird thing about a lot of this, and I've told this to Jill, my wife, is that, this whole concept of the endosome and recirculating virus and recirculating proteins, and it's a fascinating topic that at the end of the day is going to add a lot, I think, to our understanding of a lot of other things in health and disease that we didn't even expect. That's the really cool thing about a theory like this. It explains something horrible, but hopefully that horror will end. But on the other hand, I think the cool thing about it is that it explains a lot of stuff you wouldn't have expected. That's what a good theory usually does. But anyway, go ahead. I'm sorry.
We seem to be in a really interesting time of information sharing. You mentioned that some of the processing of the virus are completely open source and people are putting information out there for their peers at a faster rate than ever before.
Yeah.
And so on one hand, that feels like it's an amazing positive, that there's kind of less territoriality because everybody's trying to solve the same problem. On the flip side, the rabid attention of the media to not only fill a 24-hour news cycle, but get that information out means, as you said with the paper today, they're picking up on a little thread and missing some of the larger issues.
Yeah.
It's got to be a fascinating time to be doing this. What's giving you hope and what is causing you frustration?
Well, I'm hopeful because the truth will always come out. And I told this to someone a little bit earlier today before I came on. I was on a phone call with somebody. And I think that, what I said was, they said, oh no, this is terrible. People are going to be afraid and whatever. They see this in the media. The media is misquoting stuff. And I'm like, look, the truth will always come out. It's just a matter of time. It always wins in the end. The truth always will win. It's just a matter of how long it's going to take us to get there. And whether I'm right, whether I'm wrong, that's not the point. The point is that you need to have things that you can test that have a yes or no answer, a clear hypothesis that you can use to predict something else. That's number one. Number two, unfortunately, in the rush for information, we just throw papers up on the internet that haven't, again, as I said before, that haven't been peer reviewed. And it's tough. It's hard. You don't want to wait around. You want your idea out there. You want to tell people. It's of the best of intentions. It's not that they want to just suddenly get out there, and be famous. That's not, some people do, but the most of us don't. But the problem is that the function of peer review is really important. It's really important for guys like me or whoever, medical physicians, academic physicians, scientists, to be able to look at something and go, guys, can you answer this question for me? It kind of seems a little weird, and do that in private before it makes the public domain. And the problem is that I think the news media, kind of with the best of intentions in many cases, and probably all, they want to do good. They want people to be informed properly. Just one thing that's interesting in the 1918 pandemic, it was actually the opposite. That if you go back and look at the history of the flu pandemic, the government and all the press said, oh, things are great. No one's going to get sick. They were the opposite. And then when people were dying right and left, people were going, well, you're not telling us the truth. So it's really hard to strike that balance in a pandemic, between truth and non-truth. And I think it's hard. And, the media wants to pick up on any thread. They somehow can help a population that's frightened of the unknown and not knowing what's going to happen. And I think that, I don't blame anybody. It's the best of intentions. They're just trying. It's not so much to fill a news cycle either. I really, or to get ratings. I think at the end of the day, everybody just wants to be helpful. And, and sometimes, it doesn't come out the way it should. That's the way I look at it.
So a question that we are fortunate to have from someone who is listening, talking about, just clarifying how this is going to play out. So if I've understood correctly, if some of these theories hold water, if they're true and that's how things play out, you're anticipating that people will continue to get sick, probably at the same rate we're currently seeing, but that the degree of sickness, that the life-threatening nature is going to start to attenuate over time.
Both. I think not only will there be fewer infections because the virus will not be as transmissible, it'll be like the California virus and less like the New York virus is the best way to talk about it. It'll be less transmissible, number one. Number two, when you get it, you should get less sick. And in fact, to be honest, this paper that from this Los Alamos group looked at Sheffield, England. And while the infection rate of the virus looked to be the same, the number of people in the ICU was it wasn't statistically significant, unfortunately, because they didn't have enough people. But it looked like it numerically was about 2.5% with a good one and about 6 or 7% with the bad one. So the thing is that the prediction is that you'll see infections. You'll see people who are positive for the virus. Some may get sick, some may not. But the hopeful thing is that less will get sick, less will end up in the ICU, less will end up in the hospital, and thankfully fewer will die. That's kind of what you're trying to look for here. And does that answer your question?
Absolutely. And so
Yeah.
as that happens, then it will be not necessarily easier, but it will be safer for people to then go about, I don't know, I hope it's not a return to normal, because I'm not entirely sure what we were doing before this was the best thing for society, but to our new normal, to being back together, to being, as you said, doing human things in human spaces. It'll be safer to do that as this goes on.
Humans are human. Okay, we're human beings. We do things that make us human beings. And I think that it's hard for us not to be human. It's hard for us not to hug each other. It's hard for us not to touch each other. It's just hard. We're human beings. It's like, we go back to what we were. And I think the issue is that as much as we try to kind of keep that out of us, I think it's our instinct. It's, again, now we're talking about philosophy. This isn't just my view. This is not, who knows if it's right or not, but we want to be human beings. And I think that it's very hard for us not to be. And I think we're going to return back to the way we were. And, I think that whatever normal it's going to be, there'll be fear, and I think people will be cautious for a little bit. But again, just observe the world around you. Okay. Look, use your own, senses of what's going on. And I think your own intuition, and use that. And I think that, again, people with the best of intentions are trying to inform you, but know that there are other ideas that are out there and other ways of explaining it that may actually be right or wrong. And, I just want people to know that it probably, in my opinion, it probably will, maybe not tomorrow. I think we have to be cautious. Everything people are doing is the right thing to do because you just don't know right now. But on the other hand, my hope is that we're going to go back to being human beings and being human and doing all the things, going to sporting events, going to plays, going to graduations, seeing our grandchildren. That is going to come back. And I think that anybody that tells you no, I think we need, and the people that are saying kind of no to this, I think, again, that's between us doctors. We got to talk about it amongst ourselves a little bit and kind of come up with a thing that we can tell everybody that we all agree on. I think that's what we really have to do as docs. We're not politicians. We're not setting policy. That's not our role. Our role is really to at least give people a framework and let them decide if they believe it or not. And just the hope is that, again, we take politics out of this. It's political. It has to be because it involves political things, making people stay home. But on the other hand, that's not our role. Our role is to give people an idea, and hopefully an idea that most of the medical community can stand behind, and we have to fight about it. And unfortunately, the fight's going to be in public. It's just the way it is. It's like making sausage, but on the other hand, I think that we're going to come to an idea of what we think was happening here, all of us as physicians. And I think that, what we'll do when that happens is then everybody can figure out what they want to do with that, and everybody in their own personal mind is going to have to make that decision.
Right. The million or billion, or in this case, $100 of trillion question. Are there enough parallels to SARS or to other viruses that have followed this pattern that a time frame might be predictable? Can we look to the past to know where we are headed, or is this too different?
So if we look at SARS started in November 2003. It ended in July 2004. Okay, the disease this is most like is SARS. It's clearly like SARS. Genetically, the virus looks almost probably 90%, maybe even a little bit more like SARS. And I think the thing is, it looks a lot like SARS. And for that reason, the hope is it's like SARS. Now, the difference between this and SARS is that it's spread a lot more widely than SARS, and it could be different. I think in terms of its genetic variability, the fact it's been around, it's encountered more people. And again, remember, when you're a virus, you encounter somebody, we're not all identical, just like the virus isn't identical, we're not all identical. And it's really interesting. They did this experiment in Australia in the 50s. What happened was all these, Australia, if you've ever been there, they're very strict about quarantine. And basically what they did is that European rabbits got into Australia in the early 1950s, and they infested everything. And so these biologists said, okay, what we're going to do is we're going to release this new virus. It's called MXYV. And I've been driving my kids because I wanted to make an MXYV t-shirt, but that's besides the point. So they don't want to make it. I want to make a picture of a bunny rabbit with an MXYV t-shirt. I think it'd be kind of cool, but well, that's a whole other issue. So, so the thing is that what they did with this virus is they released it, and it was a really virulent virus. It killed all the rabbits very quickly. But what happened over a very short period of time is that it attenuated, and that the rabbits got sick, but they stopped dying. And so these biologists went really famous experiments that were done over about 30 years, and look at the virus and look at the rabbits. And it turned out that the rabbits figured out their immune system, or the bunnies themselves, had genetic variability. And they figured out, based on the variability of the bunny rabbits, whether they die or not. Then over time, the virus adapted back. And there'd be pockets where it would come up where it would kill certain animals. And it turned out there were multiple mutations in the animal and in the virus that had to interact. And the virus would try to figure it out, and then the animal would figure it out, and it's kind of they called it the arms race. It's still going on. There was a paper that was published about 5 years ago about it. What that means is that, again, this is a virus that spread, back to the original question, this is a virus that's spread all over, everywhere. And, a lot more than SARS. SARS was really limited, to be honest with you, to like Hong Kong, and it was in Guangdong province in China. And so the issue is that it was really only very limited. Only 8,000 people got it. I think 800 died or something. So it's not like this at all. And so the point is that because this is spread so far, and so wide, even though it's a lot like SARS, and we think that a lot of these mutations may be like SARS, it's hard to know at the end of the day what's going to be 100% like SARS. That's the big mystery right now, Aaron. It's a great question. We just don't know. And I think that, we're going to have to see, and at least look out and kind of watch, at least with the hypothesis, to see what happens to people as social restrictions are gradually let up. And again, my hope, I'm an oncologist, Aaron, I'm an optimist, I have to be. And, my hope with this is that it's going to hopefully be a lot like SARS and gradually attenuate away. I mean, that's what I hope.
One of the comments we received on Facebook that I want to pass along, as it echoes mine as well, is that there is great comfort and hope in this conversation as we better understand how the virus works. We have a better picture of what could be coming, and I appreciate your opening those doors. I want to make sure you get the last word here, some final thoughts, what people should be doing or thinking, or where you're headed next in your research.
I think that, again, in our research, we have a paper that, we put up that hopefully will go a little bit more way of explaining maybe what's going on, and, and we'll see if it gets published. It's not, can't talk about it too much because it's still in review, but, there's another paper that talks about how a drug that I'm very involved in, again, I don't want to tell people about specific drugs because I want them to go buy them, but, a drug that I've been very involved in my career may actually help out. It's a very cheap drug, believe it or not. Not hydroxychloroquine, although that does help a little bit, believe it or not, despite all the nonsense about it. Things like Tagamet may help, believe it or not, but don't go out and run out and get them, please, because we don't know 100%. There's a trial at Northwell Hospital in Long Island where it's going to come out pretty soon, and it'll probably give us some answers on this in the next week or two, and so the bottom line is that I want people to know there's stuff that probably will work to help them. We just aren't 100% sure of letting people know that yet because we have to do the trials right. Number two, there's a decent theory potentially about what's about to come, hopefully a good one. It's not all gloom and doom, and I think there may be some ideas, at least now that we're kind of as a group of physicians and scientists getting an idea about this thing, at least an idea. And this idea, the key that for any idea we have about this, that it be easily testable. That's really the important thing, that you want a hypothesis that you can test pretty quickly and easily. And all the things I've told you can be tested quickly and easily. And we can know one way or another. If they're not right, okay, we'll adjust them and try something else. But at least we have a place to start. We're not flailing anymore. That's all I wanted to do with this. And hopefully people get and understand that. Just be cautious, especially older people that are listening to this. I really don't want you to leave yet. Please don't walk outside, because older people, because of a lot of the things that are involved with the biology of this virus that we need to talk about tonight, are a little bit more susceptible, for a variety of reasons. And, there are various people who have certain genetic, probably predispositions, people with really high blood pressure or really type 2 diabetes that's really out of control. I think that these, just be careful. I'm telling everybody to be careful right now. But the point is, I wanted to bring across hope. Okay, this is something that I think we're starting to understand that hopefully we'll be able to deal with in a reasonable amount of time and not have to paralyze everybody for a long period of time. So that's kind of how I'd like to end.
Though our physical actions have to remain the same. You have given me a lot to think about and have uplifted my soul, and I'm sure it's true for many who have joined us this evening. Dr. Adam Brofsky, thank you so much for your time. Stay safe.
You're welcome. Thanks for having me, Aaron. I'm glad to be here. Thank you.